Hey everyone, and welcome. Today, we're diving into something truly groundbreaking, a brand new atlas of the human brain. And let me tell you, this isn't just any map. It's a high-resolution blueprint that could completely change how we fight neurodegenerative diseases.
So let's start with one of the biggest questions in neuroscience today. Is it actually possible to rebuild parts of the brain to cure diseases like Parkinson's? You know, the idea of cell replacement therapy, growing healthy new brain cells in a lab and transplanting them, is incredibly exciting. But to do that, to build anything right, you first need a perfect blueprint.
All right, so let's get into it. For years, the big dream for treating Parkinson's has been to replace the specific brain cells that are lost. It sounds so promising, right? But as it turns out, scientists kept hitting this major and for a long time, kind of invisible hurdle.
The cells we're talking about are called midbrain dopaminergic neurons. Let's just call them MDA neurons for short. Think of these as the little dopamine factories in our midbrain that are essential for controlling movement. In Parkinson's, these are the exact cells that die off. So the goal for any cell therapy seems pretty straightforward. Just make perfect replacements for these MDA neurons.
But here's where that hidden hurdle comes in. The goal is to create a pure, perfect batch of these MDA neurons. The reality, though, for a long time, what lab protocols were actually producing was, well, a mixed bag. They were getting some of the right on-target cells, sure, but they were also getting a whole bunch of off-target cells from totally different parts of the brain, like the forebrain or hindbrain. And that kind of contamination makes therapies not just less effective, but potentially unsafe.
So, how in the world do you fix a problem that you can't even properly see? Simple. You create a way better map. And this is where this incredible new framework called BrainSTEM comes into the picture. Researchers set out to build an unprecedentedly detailed atlas of the developing human brain, something that could serve as the ultimate reference point.
And when I say detailed, I am not kidding. They analyzed the genetic fingerprints of nearly 680,000 individual cells from a developing brain. I mean, that scale is just massive. It's what allowed them to create this super comprehensive map of almost every cell type, tracking exactly how they grow and who their neighbors are.
The strategy they use to map everything is it's brilliant. It's this two-step process that honestly you can think of it just like using Google Maps. First, you get the big picture view. You map your lab-grown cells against the entire brain atlas to see which city they belong to. Are they forebrain, midbrain, or hindbrain? Then you zoom way in. You take just the cells that landed in the midbrain city and compare them to a super high-res atlas of just the midbrain. That's how you find their exact street address.
So, with this new gold standard map in hand, scientists could finally do something they've never been able to do before. They could accurately grade the quality of existing lab-grown cells from protocols being used all over the world.
And the results? They were staggering. They finally saw that hidden hurdle clear as day. When they started analyzing these so-called midbrain cultures from different labs, they found that in many of them, more than half the cells weren't from the midbrain at all. They were full of those unwanted forebrain and hindbrain cells. And think about what that means. For years, researchers thought they were producing a pretty high yield of the right neurons. But it turns out those numbers were often really inflated. Without a map that was precise enough to spot all the contaminants, they just couldn't see the full picture. BrainSTEM finally pulled back the curtain on what was really going on.
And this newfound precision isn't just about cleaning up the cultures in the lab right now. As the senior author Dr. John O. Young points out, this level of detail is basically a launchpad for the future. It creates the foundation we need for AI-driven models that can design incredibly specific, targeted therapies. So, what does this all mean for the future? Well, this discovery isn't just another research paper sitting on a shelf. It's a powerful new toolkit for the entire field of neuroscience.
So why is this such a huge deal? Well, first, BrainSTEM sets a new global benchmark for quality control. Second, it gives scientists that clear, data-driven blueprint to make their methods better and create purer, more effective, and safer cells. And maybe best of all, the team made the entire atlas and the mapping tool open source, so labs everywhere can now use it to speed up their own discoveries.
And that brings us right back to where we started. For the very first time, we have a true, reliable blueprint for actually building the specific parts of the brain we need to fight diseases like Parkinson's. The journey definitely isn't over, but you know what? With a map this good, it finally feels like we're on the right road.